There is a fish which is completely transparent. Even its internal organs are visible. Which is that fish? Come on, let's see in detail. This is Soumya's capsules enriched with knowledge. The transparent fish is named as Casper fish. It is a zebrafish and it is a cross between two mutant zebrafish, Necker and Roy, that produces offsprings with translucent skin and scales. Internal organs, heart, the circulatory system, brain, and spine are viewable with the naked eye. The zebrafish in general is a freshwater fish. It is a member of Minos family. It is a model organism used to study the development of vertebrates. In this regard, zebrafish are useful because the embryo is transparent and it develops outside its mother, and its development from egg to larva happen in just 3 days. Zebrafish is recognized as an ideal model organism for studying various biological functions. The following features make them as ideal model organism. They are small and robust. They are cheaper to maintain than mice. Break of daylight triggers mating in zebrafish. On the other hand, many other fish only lay eggs in the dark. Zebrafish produce hundreds of offsprings at weekly intervals, providing scientists with an ample supply of embryos to study. They grow at an extremely fast rate, developing as much as in a day as a human embryo develops in 1 month. Zebrafish embryos are nearly transparent, which allows researchers to easily examine the development of internal structures. Every blood vessel in a living zebrafish embryo can be seen using just a low power of microscope. As zebrafish eggs are fertilized and developed outside the mother's body, it is an ideal model organism for studying early development. Zebrafish have a similar genetic structure to human. They share 70% of the gene with us. Moreover, 84% of the gene known to be associated with human disease have a zebrafish counterpart. As a vertebrate, the zebrafish has the same major organs and tissues as human. Their muscles, kidney, eye share many features with human system. Zebrafish have a unique ability to repair its heart muscles. For example, if part of their heart is removed, it can restore it in a matter of weeks. Scientists are working to find out the specific factors involved in this process to see if this will help us to develop ways of repairing the heart in humans with heart failure or heart attacks. The zebrafish genome has been fully sequenced to a very high quality. This has enabled scientists to create mutations in more than 14,000 genes to study their function. The normal zebrafish is a useful model for understanding normal and cancer stem cells. But the analysis has been limited to embryogenesis due to the opacity of the adult fish. To address this issue, Richard Mark White et al. Created a transparent adult zebrafish. The characteristic adult pigmentation pattern of the zebrafish consists of 3 distinct classes of pigment cells arranged in stripes. They are black melanophores, reflective iridophores, and yellow xanthophores. The Necker mutant has a complete lack of melanocytes due to a mutation in the gene encoding the MIFTA gene. The Roy zebrafish is a spontaneous mutant and has a complete lack of iridophores with uniformly pigmented eyes, sparse melanocytes, and a translucent skin. The gene responsible for this mutant phenotype is currently unknown. Fish that are doubly mutant for nacre and roy are named as Casper fish. This specific name is given for its ghost-like appearance. The fish completely lack all melanocytes and iridophores in both embryogenesis and adulthood. Most strikingly, the body of the fish is almost entirely transparent. In female adult fish, even the individual eggs are easily observable. Researchers compared the optical transparency of the Casper lines to several commonly used zebrafish pigmentation mutants, including golden, albino, rose, and panther. Organs that can be seen using standard stereo microscopy in Casper include heart, aorta, intestinal tube, liver, and gallbladder. Brain tissue is visible to a small extent. None of these organs are easily visible in any of the other pigmented mutants studied, other than the heart and the intestinal tube in the case of Ross. Examination of the cardiac region of the Casper mutants showed that ventricular contractions can be observed, making these mutants useful for the in vivo analysis of genetic perturbations in cardiac functions. The Casper mutants is entirely viable with encrossed adults, producing large number of viable offsprings at an expected Mendelian ratios. Casper fish will serve as a useful platform for researchers interested in processes such as stem cell engraftment and homing. For cancer biologists, it opens a window onto the longitudinal analysis of tumor growth, invasion, metastasis, and angiogenesis at an anatomic resolution not readily achievable in marine or other systems. Because the adult zebrafish is well studied to genetic and small molecular based screens, the Casper fish will allow for detailed in vivo study of stem and cancer biology in a vertebrate organism with great relevance to human biology. Thank you for watching, and I'll be coming back with more interesting scientific facts. This is Soumya's Capsules enriched with knowledge.